Good afternoon, friends. In last lecture, I had discussed the different classification of bacteria, then ultra structure of bacteria along with its explanation, then functions of each and every part of bacteria. Then we will see the morphological classification of bacteria. Before that, what is bacteria? These are bacteria are unicellular, free living, microscopic microorganism and which are capable of performing the all essential functions of the life. Yes, what is the definition of bacteria? These are unicellular, free living, microscopic microorganism capable of performing the all essential functions of the life. Then next, the bacteria possesses both DNA as well as RNA. So what is DNA? That is deoxyribonucleic acid. And what is RNA? It is ribonucleic acid. Then the bacteria are prokaryotic microorganisms and they do not contain the chlorophyll or chlorophyll. Keep in mind these are what? That is prokaryotic microorganisms that do not contain the chlorophyll. Next, they occur in water, soil, air, food and all natural environment. Yes, bacteria occur in air, soil, water, food and all natural environment. Then this bacteria can survive extreme of temperature, pH, then oxygen and atmospheric pressure. This is the introduction about bacteria. Then we will see the size of bacteria. The bacteria are very small microorganisms which are visible only under microscope. Yes, bacteria are very small microorganisms and which are visible under the only microscope. Then they are having the size range in the micron. The bacteria are stained by staining reagent and these are visualized under power of magnification, that is 1000x of compound microscope. The bacteria are stained by staining reagents and then visualized under the high power of magnification, that is 1000x of compound microscope. Then the electron microscope is also used to clear visualization of internal structure of bacteria. When we see or by the use of what? That is electron microscope, it is possible to visualize the internal structure of bacteria. Are you getting? Yes, the bacteria are visualized under compound microscope under the high power of magnification, that is 1000x. And if you want to see the internal structure of bacteria, then we use the electron microscope. Then next, shape of bacteria. On the basis of shape of bacteria, these are classified as a cocci, then bacilli, vibrios, spirilla, then spirochetes, actinomycetes and mycoplasma. I am once again repeating. On the basis of shape, the bacteria are classified as a first, that is cocci, second bacilli, then vibrios, then spirilla, then spirochetes, then actinomycetes and mycoplasma. Then first, that is cocci. Cocci are the small spherical shape or oval shaped cell. In Greek, it is kokos, it means berry. Example, micrococos. Here we can see the shape of cocci. Yes, what is cocci? These are small spherical or oval cell bacteria. In Greek, it is kokos. It means what? That is berry. For example of cocci, that is micrococcus. Here we can see the shape of cocci. Then bacilli, they are rod shaped cells. Example, bacillus anthracis. It is derived from Greek word that is bacillus. It means, the meaning of bacillus, it is stick. In Greek word bacillus, the meaning of this Greek word bacillus, it is stick. In some of the bacilli, the length of cell it may be equal to the width. It means in case of bacilli, the length and width of it may be equal. The length and width is equal. Such bacillary forms are known as cocobacilli. If the length and width of bacilli is equal, then it is called as cocobacilli. Keep in mind what I am saying. If the length and width of bacilli is same, then it is called as cocobacilli. For example, Bracella. Here we can see the structure of bacilli, actual structure of bacilli. Next, that is vibriodes. Then these are the comma shaped curved rods. Here we can see the image of vibriodes or the structure of vibriodes. For example, vibrio comma. Next, spirilla. They are longer, rigid rods with several curves or coils. They having sometimes curves or sometimes coils. Here we can see the curves or coils. They have helical shape and rigid body. Spirilla having helical shape and rigid body. For example, Spirillum ruprum. Next, spirochetes. They are slender and flexuous spiral form. Here we can see the spiral form, that is spirochetes. Next, actinomycetes. They are the characteristic shape is due to the presence of rigid cell wall. This shape is due to the rigid cell wall. For example, streptomyces. They are branching and filamentous bacteria. Here we can see the branches or filamentous structure. For example, streptomyces species. Next, that is mycoplasma. They are cell wall deficient bacteria. Keep in mind, it is important for your multiple choice questions. Yes, what I am saying, that is it means mycoplasma are cell wall deficient bacteria and hence do not possess the stable morphology. They occur as a round or oval bodies with interlacing filament. Yes, what I am saying, the mycoplasma, these are cell wall deficient bacteria and hence it does not possess the stable morphology, and therefore they occur as a round or oval bodies with interlacing filament. Then we will see the arrangement of bacterial cell. Cocci are appear as several characteristics in arrangement of group. For example, in arrangement or grouping, for example, first diplococci. First monococci, it means cocci in single. Then diplococci, then streptococci, then tetracocci, staphylococcus and sarcinae. Next, diplococci. First, they split in one plane and remain in a pair. For example, Diplococcus pneumoniae. Here we can see the structure of diplococci. Always it is present in pair. Yes, they split in one plane and remain in the pair. For example, Diplococcus pneumoniae. Here we can see the structure of diplococci. These diplococci are always present in a pair. Here we can see the pairs. Next, streptococci. These cells are divided in one plane and remain attached to form a chain. It means streptococci, it is always present in chain. For example, Streptococcus lactis. Here we can see the chain of streptococci. Then tetracocci. They divide into two planes and live in a group of 4. For example, Vaphica tetragena. Here we can see the group of 4 of tetracocci. 2 are present in above and 2 are present in below. And it is always in joint. Next, staphylococcus. Cocci cells divide in 3 planes in a irregular pattern. These cells produces bunches of cocci. It means it is present in bunches of cocci, as in grapes, as like a grapes, as you know the grapes. For example, Staphylococcus aureus and Staphylococcus albus. Yes, what is staphylococci? These cocci cells are divided in three planes in a irregular manner or irregular pattern. These cells produces the bunches of cocci, as in a grapes. It means it is present in a bunch. For example, Staphylococcus aureus and Staphylococcus albus. Next, Sarcine. These cells are divided in a three planes in a regular manner or regular pattern. Keep in mind the difference. What is Staphylococci? These are arranged in a three planes. It is also divided in three planes and it is also divided in three planes. But the Staphylococci are arranged in irregular pattern and these Sarcini are arranged as a regular pattern. Keep in mind the difference between Sarcini and what Staphylococci. These Sarcini are arranged these are these cells are divided in a three planes and it is arranged in regular pattern and this Staphylococci it is also divided in three planes and it is arranged in a irregular pattern. This Sarcine, this cells produces cuboidal arrangement of group of a 8 cell, that is Micrococcus tetragena. It is a group of 8 cells. Keep in mind Sarcine, it is a group of 8 cells. Next, arrangement of grouping formed by bacilli species. First, Diplococci, then Diplobacilli, then Streptobacilli and trichomes. First, Diplobacilli. Here we can see the image of Diplobacilli. Then second image, it is the image. Here we can see the image of Streptobacilli or the structure of Streptobacilli. Our arrangement, and here we can see the trichomes of arrangement of bacilli. I already discussed the structure of bacterial cell. It shows presence of flagella, pili, then capsule, plasma membrane, cytoplasm, then cell wall, then ribosomes, mesosomes, inclusions and spores. I am repeating only the flagella. Flagella are long, slender, thin hair-like cytoplasmic appendages which are responsible for the motility of bacteria. Keep in mind the flagella is the part of bacteria which is the only responsible for motility of bacteria. It is the organ of locomotion. This flagella is called as the organ of locomotion. They having size, the diameter of flagella is 0.01 to 0.02 micrometer and the length of flagella it is 3 to 20 micrometer. This flagella are made up of protein and flagellin. This flagella is made up of what? That is protein. The name of this protein it is flagellin. The flagellum has three basic parts, that is filament, hook and basal body. Here we can see the hook, filament and basal body. The filament is a thin, cylindrical, long outermost region with a constant diameter. And this filament is attached to what? That is slightly wider hook. The basal body is composed of small central rod inserted into the series of rings. Here we can see the rings L ring, P ring and M ring. The gram-negative bacteria, it contains 4 rings. Yes, first L ring, then P ring, M ring and S ring. In the gram-positive bacteria, it shows presence of only S and M ring. So keep in mind the difference between the gram-positive and gram-negative bacteria. The gram-negative bacteria, it contains 4 rings. And gram-positive bacteria, it contains 2 rings. What are those 4 rings in gram-negative bacteria? That is L ring, P ring, S ring and M ring. And M ring. And gram-positive bacteria, it shows presence of 2 rings, that is S ring and M rings. Then flagella, it may be seen on a bacterial body as a following manner: monotrichous, lopotrichous, amphitrichous and peritrichous. as I said earlier, what is monotrichous, what is peritrichous, and what is amphitrichous. Then the bacteria are microscopic single cell organism that thrives in a diverse environment. This organism can live in a soil, then ocean, and inside the human gut. Then bacteria represent large and diverse group of what, that is microorganism that can exist as a single cell or a cell cluster. And these bacteria are sometimes harmful or sometimes it is serve as a useful purpose. Next, the descriptive term used to describe bacteria, descriptive term, and we will see the descriptive term and adaptive feature. First, psychrophile, the growth range it is 40 degrees Celsius minus 42 plus 20 degrees Celsius. Then mesophile, growth range plus 22 plus 40 degrees Celsius. Then thermophile, growth range plus 42 plus 85 degrees Celsius. Then thermoduric, endure high temperature. Then halophile, it is salt tolerant. Then acidophile, it is acid tolerant. Then aerobes, that is air or oxygen requiring. Then obligate anaerobe, air or oxygen poison. Then autotroph utilizes the inorganic material, and heterotroph it requires the organic material. I am repeating. First, psychrophile, having growth range minus 40 degrees Celsius to plus 20 degrees Celsius. Then mesophile, having growth range plus 22 plus 40 degrees Celsius. Then thermophile, having growth range plus 40 degrees Celsius to plus 85 degrees Celsius. Then thermoduric, endure high temperatures. Then halophile, it is salt tolerant. Then acidophile, it is acid tolerant. Next, aerobe, that is air or oxygen requiring. Then obligate anaerobe, that is air or oxygen poison. Next, autotrophs utilizes the inorganic material. And next, heterotrophs it requires organic material. Next, we will see the classification of bacteria on the basis of staining, on the basis of shape, on the basis of growth and nutrition, then on the basis of flagella, presence or absence of flagella, and on the basis of motility. Next, classification on the basis of staining. First, gram staining, in that first gram positive bacteria and gram negative bacteria. Then acid fast staining it is, and third Albert staining. Here we can see the examples of gram positive and gram negative bacteria. Gram positive, cocci and bacilli. In cocci it is streptococcus and staphylococcus. In bacilli it is bacillus, clostridium, corynebacterium, listeria and actinomyces. Then in streptococcus it is beta hemolytic, alpha hemolytic and peptostreptococci. In beta hemolytic, in group A it is streptococcus pyogen. In group B it is streptococcus algaceti, then Facella and bovis. Then in alpha hemolytic it is pneumoniae and viridum streptococci. Then peptostreptococci, for example peptostreptococci magnus. Then In Staphylococcus, here we can see the flow chart of example of gram-positive bacteria. Coagulase Staphylococcus aureus, that is coagulase plus Staphylococcus aureus, and here coagulase minus Staphylococcus epidermidis and Staphylococcus sparophyticus. Then bacilli, the Clostridium, Bacillus, Clostridium, Corynebacterium, Listeria and Actinomyces. Then we examples of gram-negative bacteria, cocci and bacilli. Here the Neisseria meningitidis and Neisseria gonorrhoeae. First enteric, then non-enteric, respiratory and animal source. Neisseria, then Salmonella, Shigella, Vibrio and Campylobacter. Then non-enteric, it is Klebsiella, Proteus, Pseudomonas and bacteroids. Then respiratory in Haemophilus, Bordetella and Legionella. Then animal source Brucella, Francisella, Pasteurella and Yersinia. Here we can see the difference between the gram-positive and gram-negative bacteria. This gram-positive, this practical, that is staining on the different techniques of staining we will see in theory as well as your practical. Here we can see the difference between the gram-positive and gram-negative bacteria. This gram-positive bacteria, from this image we can conclude. Yes, what you can conclude? That is gram-positive bacteria, it shows which color? That is violet color. And gram-negative bacteria, it shows presence of which color? That is gram-negative. It shows presence of pink color. That is gram-negative bacteria. Yes, we will see the difference between gram-positive and gram-negative bacteria. First characteristic, that is gram reaction. It retain the crystal violet color and stain dark violet or purple color. Then gram-negative bacteria, it shows it can be decolorized to accept the saccharine and stained red. Then peptidoglycan layer, in gram-positive bacteria it is thick layer or it is multi-layered, and in gram-negative bacteria it is thin layered or single layered. Then tetroic acid, it is present in many, or in gram-negative bacteria it is absent. Then outer membrane, it is absent in gram-positive bacteria and it is present in gram-negative bacteria. Then lipid and lipoprotein content, it is low, that is acid-fast bacteria having lipid linked to peptidoglycan, and lipid and lipoprotein content, it is high in gram-negative due to the presence of outer membrane. Then lipopolysaccharide, that is LPS, it is virtually none, and in gram-negative bacteria it is high. Then flagellar structure, in gram-positive bacteria 2 rings in basal bodies, and in gram-negative bacteria it is 4 rings in basal bodies. Then toxin produced, it is in gram-positive bacteria primarily exotoxins, and in gram-negative it is endotoxins. Then resistance to physical disruption, it is high in gram-positive bacteria and it is low in gram-negative bacteria. Then cell wall disruption by lysosome, it is high in gram-positive bacteria and it is low in gram-negative bacteria. Then resistance to drying, it is high in gram-positive bacteria and it is low in gram-negative bacteria. Next point, that is inhibition by basic dyes, it is high inhibition in gram positive bacteria and low inhibition in gram negative bacteria. Then next, susceptibility to anionic detergent. The gram positive bacteria having high susceptibility and gram negative bacteria having low susceptibility. These are the different characteristics points of gram positive and gram negative bacteria. What I am saying, first, that is gram reaction, second peptidoglycan layer, third teichoic acid, then periplasmic space, then outer membrane, lipopolysaccharide content, that is LPS, then lipid and lipoprotein content, then flagella structure, then toxin produced, resistant to physical disruption, then cell wall disruption by lysozyme, then distance to drying, inhibition by basic dyes, and susceptibility to the anionic reagent. This is the difference between the gram positive and gram negative bacteria. Here we can see the structure of a cell wall of gram positive bacteria and cell wall of gram negative bacteria. Yes, the cell wall of gram positive bacteria, it shows presence of lipoteichoic acid, peptidoglycan, and plasma membrane. Then here outer lipid membrane and peptidoglycan and plasma membrane. Yes, this is all about the different classification of bacteria or different ways of classification of bacteria and structure of bacteria. Okay, I will stop here.